All right, so today's lesson we are going to look at genetic engineering, and we will also look at artificial versus natural selection. So just starting off by talking about genetic engineering, this is when scientists can basically take genes from one organism and put it into the genes of another organism. So it's an artificial introduction of genes from one organism into the genetic material of another organism. For example, bacteria are used to genetically engineer medicines such as insulin. Insulin is used for patients that have diabetes. So what happens is scientists will take the human insulin producing gene and they will insert it into the bacteria's DNA. And because bacteria are able to reproduce so rapidly, these bacterial colonies will then produce insulin very quickly and cheaply. So this technology called biotechnology is using or modifying living organisms to make marketable products such as insulin. And it will involve or can involve genetic engineering. Here is an example of something called transgenic. So we're still looking at technology and variation, and we're still looking at genetic engineering. So here, when we talk about transgenic, this is an organism that has been produced by moving DNA from one organism to another to create a new genetic combination. So here, in this example, human genes are added to the fertilized eggs of an animal. So we have the human gene. Here is, this was the egg donor. It's a fertilized egg here. And this human gene is added to the fertilized eggs of the animal. And then this fertilized egg is then introduced back into the female, into the womb. And then we have development happening inside of that host. And then we have offspring that develop. And this offspring now will have that human gene. So the ability to produce human proteins can then be inherited by this offspring of the transgenic animal. And here, the proteins can then be collected in the mammal's milk so that the animal doesn't necessarily have to be killed to get those proteins. All right, so that is a transgenic animal. All right, next, how has genetic engineering become important in food production? So this example is aquaculture, which is fish farming. So there has been steady decline in natural fish stocks in oceans and lakes. So the use of genetic engineering, genes have been added, for example, for disease resistance. Growth hormone genes can be added to fish eggs to increase their size and growth rate of the fish. Another gene, an antifreeze gene from Arctic flatfish, are added into Atlantic salmon and halibut. And basically what this does is it produces a protein that prevents the fish's blood from freezing, which basically allows Canadian fish farmers to continue their fish farming throughout the winters. Now, a downside to genetic engineering and fish, for example, is that natural fish that are living in the oceans, they might not be able to compete with this transgenic kind that have been altered by humans. Looking at this picture here, both fish are shown at 18 months of age. The one on the top is genetically altered. It has a length of 24 inches, 61 centimeters, and it weighs 6.6 pounds or 3 kilos. This one here at the bottom is the normal, or the one that has not been genetically altered. So same age, still 18 months old. Its length is 13 inches or 33 centimeters, and its weight is only 2.8 pounds or 1.3 kilos. All right, so because of this genetic engineering in fish farming, salmon can reach adult size in 16 to 28 months instead of 36 months for regular Atlantic salmon. Also, these transgenic salmon, they eat 25% less feed and are about 20% more efficient at converting their food into flesh.